So ENHANCE-3 study is a large phase 3 randomized clinical study that investigated the combination of azacitidine venetoclax with or without magrolimab, a CD47 targeting antibody, in patients with newly diagnosed AML who are ineligible for intensive chemotherapy. So magrolimab is a CD47 targeting immunotherapy. It was showing strong clinical data on phase 1 clinical trials when it was combined with azacitidine and also with azacitidine venetoclax in a broad subgroup of newly diagnosed AML patient population, including those with high-risk mutations such as TP53 mutation. So because of that, a large randomized clinical trial was conducted, which was the ENHANCE-3 study, where 378 participants were actually randomized between azacitidine venetoclax with magrolimab or azacitidine venetoclax with a matched placebo. These patients were treated according to the protocol and they were followed for outcomes. The primary outcome was the study was overall survival. Even though we had a lot of expectations, unfortunately, the study did not meet its primary endpoint, which is the overall survival, which was not statistically significant between the study arm versus the control arm. Unfortunately, there were also some notes about increased risk of fatal adverse events in the study arm. And there were many modifications that were done to the protocol during the course of the study. But we'll have to see how some of them may have influenced some of the risk of adverse events in those patient population before and after those ones. It was a largely highly expected study. It's a bit unfortunate that we did not have a positive result on the study in terms of overall survival, but it's certainly still an important study to help us understand what are all the key aspects that people need to be aware of when they try to utilize immunotherapy in a disease such as AML, and more CD47 targeting with potentially different agents are also possible. We'll hopefully have more information come out during the manuscript stage of this study, but it is a very important study for the AML patient population.